Hi there, my name is MacKenzie. I'm a geneticist and pediatrician at the Children's Hospital in Ottawa, Canada, and I've had the distinct pleasure and honor of working on spinal muscular atrophy research for the last 2 decades, working closely with FightSMA, the organization which has done so much in advancing the cause. FightSMA is a small charitable organization, but is characterized by a strategic and important placement of research funding in labs, not only in the United States, in Canada, and in Europe as well. And I'd like to take some time just going through some of the, really what I view as milestones in their work in advancing the cause, right from the mapping of the gene in the early 1990s to where we are today with so much promise. Back in 1991, when the SMA gene was first cloned by the laboratory of Conrad Gillum, as well as that of Judith Melke, FightSMA became initially involved, giving to the Conrad Gillum lab a DNA sequencer. Using this DNA sequencer, Conrad Gillum's lab, following their mapping in 1991, published very important, what we call mapping work, showing exactly the locale where the SMA gene was located. Although theirs was not the lab that ultimately cloned the gene, this was Judith Melke's Paris lab that did this, the work funded by FightSMA was absolutely critical in the isolation of the gene. After you isolate a gene for a genetic disorder, it then becomes an important question of how the gene works and how one goes about studying new treatments. Once again, FightSMA was there funding the generation of what they call animal models, taking the genetic defect and putting it in different species. Thus, they were the first to support the funding of Judith Melke, the creation of a novel SMA mouse model, as well as the flatworm, known as C. elegans, the zebrafish, and more recently, pioneering development of a swine model, a pig model of SMA taking place in Missouri. This is going to be important for studying new treatments, isolating tissue, and understanding how the disease works. In SMA, there's also the presence of another gene besides the SMN1 gene, which causes it, of a second gene called SMN2, which makes less of the SMN protein, but still makes some of it, and if we can learn how to turn it up, will be a therapy. FightSMA has been a pioneer in funding labs that identify novel so-called SMN2 inducers, aminoglycosides, P38 inhibitors. They go by a variety of names. The work taking place in the labs of Dr. Chris Lorson in Missouri, Dr. Arthur Burgess in Ohio, my laboratory. They really have been important in identifying novel classes of potential SMA therapies. In 1996, FightSMA co-hosted with NIH a meeting into which they invited Adrian Craner, a world leader in something known as RNA splicing. This really brought Dr. Craner over into the world of SMA, and he's been an important researcher in the field ever since, identifying novel means of therapy. An assay developed by the laboratory of Elliot Anderfee in Boston, funded once again by FightSMA, has been an important first step in the world's largest high-throughput screen of over 1 million compounds for SMN2 inducer. In addition to drug therapy for FightSMA using pharmacologic agents, one also talks about gene therapy, where one identifies a snippet of corrected DNA and introduces it into the cells. Once again, FightSMA led the way in funding both the laboratory of Arthur Burgess in Columbus, Ohio, as well as scientists in Oxford, England, showing for the first time how viral therapies may be effective in mouse models. In addition to pharmacologic or drug therapy for SMA, one can also think about gene therapy, where the actual snippet of DNA which is mutated is placed into the motor neurons. Once again, FightSMA led the way here with grants both to scientists in Oxford, England, as well as that of Arthur Burgess in Columbus, Ohio. And they went on to show that lentivirus therapy in severe mouse models of SMA had a meaningful impact. Based on this pioneering work, Brian Casper, also in Columbus, Ohio, has got some truly exciting work showing that you can actually cure SMA, severe SMA in mice if you give the drug, if you give the gene therapy early on. This is a critical question that needs to be answered. Can one really treat SMA effectively if you treat early enough? And it seems that the answer here is yes. The other exciting but technically challenging area is that of stem cell therapy for SMA. Once again, FightSMA was there funding Dr. Douglas Kerr's work at Johns Hopkins looking at stem cell therapy for severe SMA in mice. As well, FightSMA has been funding Dr. Nick Boulos in Georgia in his work on how one actually would administer either stem cells or gene therapies to spinal cord in primates, ultimately children with SMA. And one of another first, they have been funding Dr. John Bach in New Jersey's study of the so-called autonomic system to understand the severity of this problem and what might lie underneath it. In addition, the whole respiratory approach to infants and children with SMA is a fairly controversial one, once again with Dr. Bach, with Brian Weaver, pioneering an aggressive and we think appropriate approach to children with SMA. They have received support and have been part of the Thriving with SMA panel that has been a characteristic of the meetings that happen every April in Washington. Both respiratory, orthopedic, as well as the dietary approach to SMA have been topics for this panel, and ones that FightSMA have been watching closely and trying to push forward in the national agenda. In a real significant first, they really showed the way of how to do drug trials for SMA in the placebo-controlled trial of gabapentin that was conducted throughout the United States back in the mid to late 90s. Although the actual trial was showed that the gabapentin did not have an effect, it was a very important template for how to do clinical trials for spinal muscular atrophy, and has really served as a role model for other trials that have followed since. They're also squarely behind Kathy Swoboda's trial of children who've been diagnosed, infants who've been diagnosed with SMA prior to any symptoms, seeing whether sodium phenylbutyrate makes a difference. And this is really a critically important trial because these infants have not started to lose the motor neuron, and therefore we'll be able to tell whether some of the agents that she is looking at are effective. In general, a theme of good citizenship is something that FightSMA strives for. Their supportive meetings, their annual meeting in Washington, meetings such as the SMA Drugs Summit, supporting the standard of care statement that was issued recently, have all been part of their portfolio. As well, the patient registry has an important aspect of what we do. Common resources, things that can be generated by one lab but used by other labs, is another recurrent theme that FightSMA has tried to achieve. For example, the so-called SMN ELISA, the ability to actually measure the levels of SMN protein in a robust and high throughput manner, enabling drug screens, pioneered in Glenn Morris's lab in Wales, has been something that we put seed money into, as well as the dissemination of the iPS stem cells discovered by Clive Svensson and FightSMA scientific director Chris Lawson recently. Allowing these cells to be spread to other laboratories has been another project that they have supported. One of the most exciting things with our SMA research is its unanticipated consequences for other disorders. Work in my laboratory, for example, has real application to a disorder known as myotonic dystrophy. Using techniques that we really discovered through our work on spinal muscular atrophy has identified what we think is an effective therapy for this really common form of muscular dystrophy.